KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Education Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing asexual reproduction in plants and animals. Asexual reproduction in plants as well as in animals. So in previous video, exclusively we have seen. So what is the information we have seen now? So we have seen information regarding agamospermy and spores from agamospermy we have seen exclusively. Now in this video, I will give information regarding spores formation. So I will give information regarding spores formation. So actually asexual reproduction in plants, how many types are there? 3 types are there, no? So in previous video agamospermy I have given explanation. Now in the present video I will give explanation of spores formation. Already in previous videos, so I have given information regarding vegetative propagation. So thereby after completion of this spores formation, I will give you explanation in asexual reproduction in animals. Are you getting? So thereby asexual reproduction in animals later I will explain. So once that particular asexual reproduction in animals is completed, so definitely I will enter into the sexual reproduction in animals. So right, now let us start second side heading, that is spores formation. Where? In plants. Asexual reproduction in plants. So there spores formation point of view, fungi and simple plants such as algae reproduce through the spores. Spores after certain time gives rise to young ones. So this particular point already in previous video also we have discussed. So thereby what is the point? So spores formation point of view. So fungi and simple plants. So the fungi and simple plants such as, what are those simple plants? So for example algal members reproduce through spores. And spores after certain time gives rise to young ones. So what are the spores here are produced by the or from the fungi or simple plants, for example algal members. All these spores, so germination takes place after certain time and gives rise to young ones. Next one, in different organisms, the spores are of different kinds. So here in definitely spores will be like morphologically will be very different in different organisms. So means in all organisms spores structure is not similar, that is dissimilar we are learning. So what are the kinds of spores are there in different types of organisms? So let us start to discuss one by one. So first of all, how many types are there means once you see there, zoospores, sporangiospores, chlamydiospores and chlamydospores, and fourth one is conidia. One more time, zoospores, sporangiospores, next one chlamydospores, next one is conidia. First one, zoospores point of view, what is the information? Zoospores are nothing but a spore of a certain algae, fungi and protozoans, so capable of swimming by means of flagellum. So means these zoospores are nothing but spores These zoospores are nothing but spores only. So these kind of spores development we can see in algal members, fungal members and protozoans. And what is the important or what is the specialty of these zoospores means these are all capable of swimming, so they can swim. So by the nature of swimming they cannot move from the one place to another place. Sir, how they will swim, sir? But so what is the what is the system helps in swimming means the structure is nothing but flagellum. So here because of the presence of a flagellum, flagellum singular, flagella plural. Okay, so that particular point also you can save in your mind. So thereby definitely zoospores are having a capability to transfer from the one place to another place, so they can swim. So they have the capability to swim. So by the having that nature of the swimming, so it can move from the one place to another place. So it can swim with the help of flagella. Flagella is a plural number. So many number of flagella we can be taken as a flagella. Am I right? Now zoospores we have completed. Zoospores formation we can see in algal members, fungal members and a few protozoans or simply protozoans. So here these zoospores has the capability to swim, so by means of flagellum. Next one is sporangiospores. Sporangiospores point of view, these sporangiospores are produced inside of a specialized structures known as sporangia. These are sporangiospores. Sporangiospores are produced inside of a specialized structure or specialized cells. So here the specialized structures I have written. So specialized cells also you can take. Okay, specialized cells or the structures are known as sporangia and remain enclosed in the structures until maturity. So means when these specialized sporangiospores are produced in specialized cells or specialized structures, these sporangiospores will not come out from the sporangia. So when they will come out, once the sporangiospores are matured, then only these sporangiospores will come, or these sporangiospores will come out from the sporangia. Until the maturity or till the maturity, these sporangiospores will be there within the sporangia. Are you getting? So sporangiospores are the spore-like structures are developed inside the specialized structures. Inside the specialized structures or the specialized cells are known as sporangia and remain enclosed in the structures until maturity. Right. Next one, third one, chlamydospores. Third one, which one? Chlamydospores. So in chlamydospores, what is the information we can save means? So there, chlamydospores point of view, it is a thick-walled. Chlamydospores are thick-walled, large resting spores of several kinds of fungi, including Ascomycota, their example Candida, and Basidiomycota, example Pannus. So one more time, chlamydospores are thick-walled large resting spores. Thick-walled large resting spores. What is the meaning? No. So these spores will be like or will have thick-walled outer layer because of the thick-walled outer layer and resting spores. These are all spores in unfavorable conditions. Take rest. Or takes rest. So that's why resting spores of several kinds of fungi. Several kinds of fungi include Ascomycetes. So we know very well Ascomycota we will call Ascomycetes and Basidiomycetes. So means in Ascomycetes as well as in Basidiomycetes, thick-walled resting spores if you find means. So these thick-walled resting spores, these thick-walled resting spores definitely we can call them as chlamydospores. Point you understood? No. So these chlamydospores for our convenience to study we can see in Ascomycota and Basidiomycota or Ascomycetes or Basidiomycetes. So best examples in Ascomycetes also where we can see means in Candida, and in Basidiomycetes we can see in Pannus, P-A-N-U-S. Next one, third one is Conidia. Conidia there. The spores produced asexually by various types of fungi at the tip of the specialized hyphae or hypha. So means here these conidia spores or these conidia spores produced asexually. That's one we are studying under asexual reproduction only. So these kind of spores means conidia spores produced asexually by various types of fungi or various fungi at the tip of the specialized hyphae. At the tip of the specialized hyphae known as conidiophores. At the tip of the specialized hyphae, at the tip of the specialized hyphae, that particular hyphae name is conidiophore. 4 1 2 3 4 likewise we will call now adi 4. Okay, conidiophore. Conidiophores. Now these conidia members means here what are the spores are produced on the tip of the conidiophores. All these spores are haploids. All these spores are haploids. That particular point also you try to say. Right. Now in plants, so let me revise this entire information. So till now what is the information we have discussed? Now let me revise. Now in plants, asexual reproduction, how many types are there? Three types are there. First one is agamospermy, second one is spores formation, and third one is vegetative reproduction. So in this video we are discussing second side heading, that is spores formation. So there fungal members as well as some simple plants, for example algal members. So there these kind of plants reproduce through the spores. So there these spores in favorable conditions definitely germinate and form young ones. Next one, in different organisms the spores are different kinds. The spores are different types are there. Zoospores are there, sporangiospores are there, chlamydospores are there, and conidia are there. Now zoospores definitely, so where we can see means the presence of zoospores we can see in algal members and fungal members and protozoans. So these zoospores have the capability, to swim so thereby they can move from the one place to another place, so by the presence of flagella. Next one, sporangiospores are produced inside of the specialized structures known as sporangia. So here these kind of sporangias or reproductive structures, within the reproductive structures sporangiospores development we can see. These sporangiospores not outside of the sporangia, produced inside, endogenous origin. And remain enclosed. So here when these sporangiospores are produced within the sporangia, so these sporangiospores on the spot will not come out, so until maturity or till maturity, so these sporangiospores will present within the sporangia. So that's why here what is the point here we are learning. So these sporangiospores remain enclosed in the structures until maturity. Next one, third one, chlamydospores point of view, it is a thick walled large resting spores of several kinds of fungi, including Ascomycetes and Basidiomycetes. In Ascomycetes best example Candida and Basidiomycetes best example Panas. Next one, conidia point of view. Conidia, so the spores are produced asexually by various fungi. Conidia point of view, so these are also types of spores only. Okay, so all these are all produced asexually by various types of fungal members at the tip of the specialized hyphae. At the tip of specialized hyphae known as conidiophores. Means on the conidiophores we can observe the presence of conidia. And these conidia members are haploid cells. These conidia members are haploids. Now this is somewhat information regarding, so which one, asexual reproduction in plants. Next one, asexual reproduction in animals. Now let us see asexual reproduction in animals. So there first one is fission. So how many types are there means? So first one is fission, second one is budding, and the third one is third one fragmentation, and the next one cyst and spores. So likewise there are various types over there, but in this video I can cover fission and fission types and budding. I can cover remaining two in the next video. Let us discuss. Okay, now asexual reproduction in animals, let us start to discuss. So their first one is fission. Fission is the commonest and simple method of reproduction. Simple method. So it is very common. So fission type of asexual reproduction, it is a very commonest and a simple method of reproduction. In fission, body of an individual undergoes division to produce two or more equal sized daughters. Try to save this point in your mind. In this fission, what is the body undergo for the process of fission? Definitely that particular individual produce two or more equal sized daughter cells. So means thereby, suppose if one particular organism going to be divided by the process of division means, so in this particular case what is happening? One particular individual undergo the process of fission and thereby to produce two or more equal sized daughters. Am I point understood now? One organism, parental organism is there, that particular organism is undergo the process of fission means one particular organism definitely divided into two equal daughter cells. That they are saying. Sometimes two or more than two. So thereby but equal sized daughters they are saying. So thereby here what is the point here we can highlight more and more means, so this particular fission for our convenience to study, now we are dividing into two types. Two types there. First one is a binary fission and the second one is multiple fission. First one is binary fission and the second one is multiple fission. Now binary fission and multiple fission, let us see one by one. Now binary fission point of view, it is one of the most common modes of asexual reproduction. Which one? So the binary fission is one of the most common type of asexual reproduction. Don't forget, right, important one. It is the most common. It is the most common mode of asexual reproduction. Which one? Binary fission. So in a binary fission what is happening? No, the parental cell divides into how? Two small and approximately equal sized individuals. Binary fission. So as we seen before, so definitely one particular organism is there means that particular parental cell divides into two small and approximately equal sized individuals. Is a rough diagram also I don't have interest to draw. Why? Because one parental cell divides into two equal halves. Why diagram? So in our mind also we can think about it. So thereby one particular parent cell is there. Finally that particular parent cell is divides into two equal sized particles or the two equal sized individuals. So this kind of a phenomenon very exclusively we can observe in bacterial members as well as amoeba members. In amoeba and in bacteria members, this kind of a binary fission we can watch, we can see. Next one, modes of binary fission. So there are different types of binary fissions are there. So there are different types of modes are there. What are those? So first one is a simple binary fission. We have taken binary fission of bacteria members and amoeba members. So deep information. Sometimes in orange color we have written so this particular information. So they are asking. So let us discuss. That's why. Right now see there. So binary fission or simple binary fission. Their best example is amoeba. Next one, longitudinal binary fission. Longitudinal binary fission best example. Longitudinal and transverse, in so many examinations they are asking. So try to find out this one. Longitudinal binary fission best example Euglena, and transverse binary fission best example Paramecium. So longitudinal binary fission, best example euglena. Next one, transverse binary fission, best example which one? Paramecium. And oblique binary fission, keratium. Keratium, CERATIUM, keratium or ceratium, whatever it is, but spelling you try to remember. So oblique binary fission, keratium, and transverse binary fission, paramecium, and longitudinal binary fission, euglena, and simple binary fission, example amoeba. So when you will watch this video, after when you will watch this one, once you close your eyes and try to recollect the examples, so then only you can remember, Sariya? Right. Longitudinal binary fission, best example euglena, and transverse binary fission example which one? Paramecium. And oblique binary fission, best example is ceratium or keratium, and simple binary fission, best example amoeba. Shall we start to move forward to learn the multiple fission? Right, let us move forward now. So multiple fission point of view, the word itself giving information. So definitely that is in this kind of a division, what is happening? Parental cell divides into many small daughter individuals simultaneously. Point you understood? In this type of division, parental cell divides into many small daughter individuals simultaneously. One parental cell is there means that particular parental cell divides not into two, that is more than two, or simply many small daughter individuals simultaneously. So this kind of example we can be seen in plasmodium. And one more here you see there, definitely all the time this kind of examples we are observing. So best examples are plasmodium, and one more is amoeba members. The plasmodium and amoeba members, it occurs, and one important point is so it occurs only in unfavorable conditions. So thereby here what is the point? Munnadi we have seen in a binary fission, we have seen the examples of amoeba as well as paramecium we have seen. So thereby here in multiple fission, so examples again we have taken plasmodium and amoeba. So thereby multiple fission only we can observe in unfavorable conditions, but simple binary fission and transverse binary fission we can observe in favorable conditions. So try to get this particular point. Simple binary fission, example amoeba, and transverse binary fission, example paramecium. This kind of a phenomenon we can observe only in favorable conditions, whereas in unfavorable conditions, whereas or adverse conditions, adverse conditions also meaning unfavorable. So thereby in that particular unfavorable conditions, so multiple fission will operate. So in that particular case, what is happening? One parental cell divides into many small daughter cells. So best examples are plasmodium, and one more is amoeba. So next one. So this is some more information here. So this particular point you try to save in your mind. Right. Next one, multiple fission point of view, in our textbook also they have given one important point. NC station. Did you remember there? What is the point? So one more important, NC station formation means one particular amoeba is there means around of that particular amoeba, so many layered or multi-layered structures of formation takes place. So there within the multi-layered structures, amoeba members are there. When favorable conditions comes, at the time definitely amoeba gives rise to many single or many amoeba members. That kind of condition we will be taking as NC station condition. So that particular information already we have seen in textbook information. So while explaining the textbook information, so we have learned Neet information point of view, so binary fission and multiple fission information. So in unfavorable conditions, so multiple fission formation we have seen now. Right. Now in asexual reproduction in animals, second one is budding. Second one is budding. In budding, what is the information we can see means budding is an unequal division. Once you try to save this point in your mind. So budding is an unequal division of the parent where the identify of the parental body is still maintained. So unequal division only of the parent, but here this particular parent so definitely is consist of that particular budding, it maintains still. So means that particular bud will not detach from the parent, that definitely that will be attached or still attached to the parental body. So that is the information. So budding is an unequal division. Budding is an unequal division of the parent where the identify of the parental body is still maintained. So next one, budding is in contrast with the or contrast to the binary fission. So thereby here what is the point? So contrast means opposite. Contrast means opposite. So thereby budding, the budding process, it is opposite. It is opposite to the binary fission. It is opposite to the what? Binary fission. So where the plant body divides, where the plant body divides equally into two and no identify of the parent is left. So means here no also is there, no identify of the parent is left, means thereby here what is the point? Budding is a contrast to the binary fission. So here even though this budding is a contrast to the binary fission, so in this budding, so the plant body is divides so in a binary fission the plant body is divides equal binary fission only. So what is the information? The plant body which divides equally to the parental body. So thereby here what is happening? No, in this particular case, no, so there is no any new characters or there is no loss of parental characters. So here somewhat information they have given related to the binary fission only. So thereby budding is a contrast to the binary fission, and where the plant body divides equally into two and no identify of the parent is lost. So there is no loss of the parental characters in the binary fission. Next one, budding is a formation. So once you see there, budding is a formation of multicellular or unicellular outgrowths. Outgrowths in brackets budding. Budding is a formation of multicellular or unicellular outgrowth on or inside of the parent. Means here this kind of budding definitely we can see on the organism or inside of the organism. Chance is there to be outside of the organism. That's why here once I have written, once you see there on, means so budding is a formation of a multicellular or unicellular outgrowth on or inside of the parental body. So thereby chance is there to be on the parental body or chance is there to be within the parental body. Next one, bud formed inside the parental body. Suppose if the bud is formed within the parental body, so suppose that kind of example we can be taken as gemmules. Suppose if the bud is formed inside the parental body, we will be taking as gemmules or statoblast. Which one? Statoblast. Suppose if the bud is formed inside the body or inside of the parental body, so best example gemmules or statoblast and formed on the external surface of the body or exogenous body, best example hydra. So thereby here in a bud, actually here gemmules, so gemmules information also we have seen. So gemmules and which one? So budding in a hydra also we have seen in our textbook information point of view. Bud formed inside of the body. So buds are formed inside of the body called gemmules or statoblast, and suppose if the formed externally, suppose if the buds are formed outside of the body or exogenous body or exogenous bud, we will be calling or we will be seeing in hydra. Next one, budding is found in sponges. Budding is found in sponges and annelids in annelids coelenterate. So annelid coelenterate, hydra, and among the fungal members, in some fungal members, so for example yeast, in yeast also we can see the presence of budding. So means here the budding, external budding point of view, best example hydra, and inside of a budding, best example gemmules or statoblast, and in annelids hydra, and yeast, in yeast also budding we can see. So thereby here, so it is smaller rough diagrams of yeast. It is yeast, koncham like outgrowths are started later. So complete outgrowth bud formation here we are observing, and this is a particular information of gemmule. So gemmules in sponge. Gemmules means here, suppose here buds are formed inside of the body, means gemmules we are observing now. Gemmules are where, so in a sponge we can observe the formation of gemmules. Formation of inside bud we will be calling as a gemmules. So these particular gemmules, asexual reproductive structures, we can observe in a sponge. So this is a particular information, gemmules in a sponge there. So once you see their outermost layer we will be calling as outer covering layer, and inner layer we will be taking as inner covering layer, and ulle motte like structures like a spherical shaped structures are there. These are all archaeocytes. These are all nothing but archaeocytes. Next one, in a line like structures are there. These are all spicules. Spicules, and within the inner side, inner covering layer surrounding the which one archaeocytes. Next one, outer layer is there, and here one opening is there. This particular opening we will be taking as micropylar region, or simply you try to remember as micropyle. So this is a structure we can observe in a sponge. Okay, so within the sponge, here presence of which one gemmules we can observe. So this kind of a diagram is there in our NCERT textbook, but labeling not given. Labeling not given. So by seeing this video you can label, so what is the diagram is there in our textbook. So this is the information regarding asexual reproduction in plants and asexual reproduction in animals. In animals up to two-sided eggs I have covered. In the next video, fragmentation, cyst and spores I will cover. Once if I complete this asexual reproduction, definitely I will start the information of sexual reproduction. So try to watch this video, so don't skip anywhere, so thereby you will get the knowledge very exclusively. So in a NEET examination definitely you will get more marks if you watch these videos clearly and carefully. Thank you, Ma.